Good morning, students. Today we will continue with our next topic, that is general characters of bacteria. In this unit, the microbiology unit, third unit, unit 3, we have microscopy. First, that is 3.1 is your introduction to microbiology, 3.2 is microscope, that is bright field microscope, phase contrast microscope or fluorescent microscope, and your electron microscope, that is scanning electron microscope and transmission electron microscope. All these will be dealt by Murali sir. And I have yesterday started outline classification of microorganisms, we have seen it. Next, today we are going to general characters of bacteria. Already you know we have completed this in your first sem only. And these general characters, the bacteria, they are first time observed, they are the naked, they cannot be seen with naked eyes, they may be in single celled form, unicellular or multicellular, they are present enormously in the environment, these bacteria, they were first identified by Leeuwenhoek. Leeuwenhoek in 1676, he has observed the presence of such type of small microscopic organisms, and later on in 1838 another scientist by name Edinburgh, he coined the term bacteria, he called them as bacteria. Next, these bacteria, coming to the terminology, what are the general characters of bacteria? The bacteria, they are first okokkati chepta nenu. The bacteria are omnipresent. First character is they are omnipresent. That means they are present everywhere in high temperatures. Next, acidity, acidic nature. Next, they are present in moisture. Okay, they are present in saline conditions. See here, that means the microorganisms, they are omnipresent, they are present everywhere, just like God. God is omnipresent, so the microorganisms are also omnipresent. They are present in temperatures. What are the temperatures? They may be present in low temperature, high temperature. At low temperature and high temperature, that is at 75 degrees centigrade also, they are present in hot water springs. They are called as geysers. In hot water springs they are present, and at low temperature in Antarctica in Antarctica also they are present, that is at freezing temperatures they are present, and they are present in hot water springs, that is at 75 degrees centigrade also they are present. Now, in acidic nature, the pH is, they can range from 0 to 11, that is pH 0 to 11, that is acidic and alkaline. That is 7 is the neutral pH, so they are present below 7, that is from 0 pH, that means high acidic condition. Ante if you go to your concentrated sulfuric acid, concentrated HCl and concentrated H2SO4, their acidic nature is 1 or 2, but here at 0 pH also they can stay, they can live, and at high alkaline condition, that is at 11 pH also they can live. Next, in moisture, no doubt in moisture in the atmosphere they are present. In saline, that is in ocean and sea, in the sea water and ocean water they can live. This is the omnipresent nature, first characteristic feature of your this one. Now coming to the second one, that is nutrition. What is nutrition? How they feed? They are heterotrophs. See here, they are heterotrophs. Heterotrophs means they cannot synthesize their own food, they are dependent on other organisms for their food. They may be parasites, saprophytes, and third one is symbionts. What is the meaning of parasite? These are the organisms which live on another organism for their food. They are called parasites. Next, saprophytes. These are the organisms which live on dead and decaying organisms by feeding on them. The dead and decaying organisms are fed by, or these are the saprophytes live on dead and decaying organisms. Coming to the symbionts, these are the organisms, they live on other organism, but mutually the organism and the microorganism, they are benefited to each, they are making a benefit to each other. That means they are benefiting each other. Example is Rhizobium bacteria. This Rhizobium bacteria is there, and this is living on in the roots of legume. When they are living on the roots of legume plants, in the roots of legume plants, the roots will give food to the rhizobium, and this rhizobium will fix atmospheric nitrogen. Okay? Atmospheric nitrogen, and this nitrogen is given to the roots of legume plants, because the atmospheric nitrogen cannot be absorbed by the roots of the legume plants. So, the atmospheric nitrogen is fixed to ammonia form, and it is given to the roots of the legume plant. That means both are benefiting each other, they are interrelated to each other, they are benefiting each other. Such type of microorganisms are called symbionts. So, the microorganisms nutrition is, they are of heterotrophs, parasites, saprophytes and symbionts. Some of the microorganisms are autotrophs also. That means they can synthesize their own food material. If they have to synthesize their own food material, they are having bacteriochlorophyll. What is this? Example for autotroph is nostoc. That is your cyanobacteria or blue-green algae. Blue-green algae, as they consist of bacteriochlorophyll for in them, in their cells. So, these are the two types of their nutrition. Is it okay? Now, coming to the next one, that is, we have seen the omnipresent nature, we have seen the nutrition. Now, we will see the size of your bacterium. The size of the bacterium depending upon the shape. First, let us see size and shape together. Okay? Size and shape we will see together. That is, third one is size and shape, or shape and size. Now, the bacteria may be cocci, that is round. Next, bacillus, they may be square shaped or spirillum, like this. Next, vibrio, comma shape. These are the four shapes of bacterium. Now, coming to the cocci, cocci is, its size is ranges from 0.5 to 2 micrometers in diameter. The diameter is 0.5 to 2 micrometers. Coming to bacillus, the length is 10 micrometers to 15 micrometers, and width is, or breadth or diameter is equal to, this is 0.4 to 1.5 micrometers. This is 0.5 to 1.5, 0.4 to 1.5 micrometers, and the length is 10 micrometers to 15 micrometers. Coming to the spirillum, its length is 15 micrometers to 20 or 25 micrometers. Okay? Next, its width is only 0.2 micrometers. అంటే ఏంటి, ఇక్కడ చాలా తక్కువ ఉంది. దట్ ఈస్ డయామీటర్ ఈజ్ 0.2 micrometers. Coming to the vibrio, length is, it is only 10 micrometers. Up to 10 micrometers, the length will be up to 10 micrometers only, and width is, or diameter is as that of your spirillum, 0.2 micrometers. This is the size of your bacterium. Now, coming to the cocci. Cocci are circular, bacilli are rectangular shaped, spirillum is spiral shaped, and vibrio is comma shaped, all of you know. Now, these cocci can be seen in monococcus, diplococcus, streptococcus, staphylococcus, Sarcina. See here, monococcus means 1, diplococcus 2, streptococcus means a chain of bacterium is there, staphylococcus a group of bacteria will be there, irregular group of bacteria will be there, Sarcina means 8 bacteria will be there, front 4 and back 4, 8 a cube like structure of bacteria will be there, that is known as, it is 8 celled structure, that is known as Sarcina. These are the different types of bacteria. The example for monococcus is micrococcus, diplococcus is your streptococcus, or you can say it as your tuberculosis bacteria. Next is streptococcus is a chain of bacteria, that is Streptococcus albus. Next, staphylococcus, a group of cells are there, that is Staphylococcus indicus. Next, Sarcina, you can give any example, that is E. coli cells also you can give. Now, coming to the cocci cells, the Escherichia coli is the best example that stays in our intestines and that helps in digestion of our food. Is it okay? Now, coming to the bacillus, it is monobacillus, diplobacillus, streptobacillus. Monobacillus example is Xanthomonas, diplobacillus example is your Pseudomonas. Coming to the your streptobacillus, the example is your Mycobacterium. The example for streptococcus, that is a chain of bacteria will be there. Ante here monococcus, diplococcus and streptococcus, that is a long chain of bacteria will be present, this is called as streptococcus. Now, coming to the spirillum, spirillum is single and vibrio is also single. They do not show monospirillum, diplospirillum like that, they are single only. And this vibrio is causing, best example for vibrio is vibrio cholera. Vibrio cholera, it causes your cholera. Vibrio cholera causes cholera disease, and spirillum, it is spiral in shape, spring shaped. Is it okay? This is the size and shape of your bacterium. Now, coming to the next one, that is we will see the appendages, that is structures that are present in the bacterium. They are, next one is your fourth one is your flagellum. Flagellum is an appendage or it is an organ that helps in the movement of the bacterium. First one. Second one, this flagellum, it consists of a protein called as flagellin. It consists of a protein called flagellin. Next, it consists of 11 plus 2 arrangement. That means inside it consists of 2 fine thread like structures which are surrounded by 11 structures, 11 fiber like structures which are surrounding it, and inside only 2 strings are there. This is known as 11 plus 2 arrangement. That is, the flagellum consists of 11 plus 2 arrangement. Now, the flagellum which is present in the bacterial cells, it is of 5 types or 4 types. First one is, it may be, see here, the flagellum may be present, it is monotrichous. Why it is called trichous? Flagellum is also known as trichon. See here, flagellum is also known as trichon, and this trichon means thread like. In Latin language, trichon means it is a thread. Thread is known as trichon, means thread in Latin language or Greek language. Now, it is monotrichous. Next, lophotrichous. Next, amphitrichous. Next is your peritrichous. Next, last one is atrichous. Atrichous. See here, this is these are the different types of flagellum, bacteria, depending upon the presence of your bacteria, flagellum. Monotrichous, it will say only one. That is, this is your bacterium. one flagellum will be present. Lophotrichus means on one side of the bacterium a bunch of flagellum may be present. Amphitrichus, amphibian means frog is an amphibian that can live both in water as well as on land. So, both, that means on both ends you can see a bunch of flagellum. Single flagella or a bunch of flagellum are present on both sides, it is known as amphibian can live both in water and on land, is it or not? It can live both in water as well as on land. So, amphi means both. So, on both ends flagellum is present. Next, coming to the peritrichus, the flagellum is present throughout the body of the bacterium. This is known as peritrichus bacterium. Now, this monotrichus, xanthomonas is the example. For lophotrichus, it is pseudomonas is the example, and for amphitrichus it is your Streptococcus albus, and for peritrichus it is, you can give your E. coli. Next, your atrichus means the bacterium is without flagellum. Example is Vibrio cholera. It is Vibrio cholerae is the example for your atrichus bacterium. These are the different types of bacterial cells depending on the flagellum. Now, coming to the next one, that is your next appendage is your pilus. See here, it consists of, next one is pilli. Pilli, or it can also be called as fimbriae. They are also called as fimbriae, and these pilli, they consist of a protein called pillin or fimbrin. It consists of a protein, it is made up of a protein called pillin or fimbrin. Now, see here, this pilli, what it will do is, these are present in gram negative bacteria. They are present in gram negative bacteria. Now, when these are present, that is, the pilli are the nothing but they help in the formation of your conjugation tubes. See here, the pilli is present, it helps in the formation. For the first one is, it helps, this is your bacteria, they consist of pilli. Some pilli are present on their surface, and these pilli, they are of two types. They may be helping for general pilli, they help in attachment to the or joining to a surface, and second one is a sex pilli, they help in the formation of conjugation tube. Suppose this is your bacterial cell, this is another bacterial cell, and these two bacterial cells, they have pilli, this also has pilli. Now, both the pilli touch each other and they produce a conjugation tube, and the wall gets dissolved, and the material gets exchanged between these two, genetic material gets exchanged between these two conjugates or these two bacterial cells during conjugation. This is known as your conjugation, where the sex pilli help in the formation of conjugation tube and helps in sexual reproduction. They help in the transfer of sex factor from one bacterial cell to another bacterial cell. This is your pilli, they are present especially in gram negative bacteria. Okay. Now, coming to the next one, that is cell wall. Next is, sixth one is, all cell organelles are absent except ribosomes. Ribosomes are present, these are of 70S type, not 80S like eukaryotes, they are of 70S type, and these ribosomes, they are 30S and 40S, subunits are of ribosomes are 30S and 40S, and they are combined together to produce 70S ribosomes. Now, it consists of a cell wall. Next one is a cell wall. that is seventh one is your cell wall. This cell wall, see here, this is your bacterial cell and this is your cell wall. This cell wall is made up of peptidoglycan and muramic acid. This you will be calling in intermediate as a murin. So, peptidoglycan and muramic acid both are combined with each other to produce your structure called cell wall. And this peptidoglycan and muramic acid, they are nothing but they are your carbohydrates. So, in gram positive bacteria, they consist of peptidoglycan plus muramic acid, and gram negative bacteria, they have peptidoglycan plus muramic acid plus they consist of peptidoglycan layer is only 10% and 80% is lipids. In gram negative, peptidoglycan layer is 10% and your lipids are 80%, and in gram positive, peptidoglycan is 80% and lipid is 10%. And in gram positive, they consist of teichoic acid. In gram negative, teichoic acid is absent. Okay, teichoic acid is absent in gram negative bacteria. Now, see here, these gram positive bacteria, the thickness of the cell wall is 25 to 30 micrometers in diameter in thickness, but here it is 10 to 15 micrometers in thickness. Okay, it is 10 to 15 micrometers in thickness. That means the gram positive cell wall is thick and gram negative cell wall is thin. Okay. Now, it consists of peptidoglycan and muramic acid 80% and peptidoglycan 10% and lipids 80%. And next one, teichoic acid is also present, teichoic acid is absent. Here lipids are only 10% and the thickness of the wall is 25 to 30 and the thickness of the wall is 10 to 15 micrometers only. Next, see here, this gram positive bacterium, when you are making the bacterial cell to stain, it produces with crystal violet, it produces purple color. When purple color of the bacterium is produced, it is known as gram positive bacteria. When you will get pink color, that is safranin is there, safranin is used, it produces pink color, and when pink color is produced, it is gram negative bacteria. That means depending upon the thickness of the and the composition of the cell wall, the bacteria are classified into gram positive and gram negative. Okay. Now, these bacterial cells, plasma membrane, this is your peptidoglycan and muramic acid, they consist of NAG and NAM, they are alternatively present, NAG, NAM, like this. Alternatively, these layers are present. What is these layer? NAG means N-acetyl glucosamine and NAM means N-acetyl muramic acid. That means the cell wall of bacterial cells consists of, the bacterial cell wall consists of alternating units of NAG and NAM. NAG means NAG means N-acetyl glucosamine and NAM means N-acetyl muramic acid. They are alternatively arranged in the cell wall, and depending upon the thickness and depending upon the composition of the cell wall, they are classified into gram positive and gram negative. Is it okay? Gram positive are always stained with crystal violet and they are purple in color, and gram negative are always stained with safranin, gram safranin, and always they are always pink in color, and they are always pink in color. Okay. Now, this is about your cell wall. Coming to the plasma membrane. This plasma membrane, it consists of protein, lipid, protein layer. It consists of protein, lipid, protein layer along with carbohydrates. And now this lipid protein layer is present, and it consists of, it shows fluid mosaic model. All of you know that. It consists of fluid mosaic model. It shows fluid mosaic model of your plasma membrane, where it consists of intrinsic proteins and extrinsic proteins which are embedded in the lipid layer. See here, lipid is present. Suppose this is your lipid layer. In this, externally some proteins are there, they are called extrinsic proteins. Internally embedded some proteins are there, they are called intrinsic proteins. Both extrinsic and intrinsic proteins are present in the plasma membrane. This is known as fluid mosaic model. Just it represents, just it looks just like icebergs floating in an ocean. These are the icebergs floating in a lipid ocean. So it is known as fluid mosaic model, because it is fluidy in nature and it consists of mosaic nature. What is mosaic? Here and there small dots are there, that are nothing but your extrinsic proteins and intrinsic proteins are there. So this is your fluid mosaic model. This consists of PLP layer, that is your protein, lipid and protein layer, and here and there this will start folding, and this is known as mesosome. And this mesosome is the infolding of the plasma membrane, and this infolding of plasma membrane, it consists of respiratory enzymes and photosynthetic enzymes. 2 minutes. Now this mesosome, it consists of both respiratory enzymes and your, if it is a chlorophyll is present. Next thing, it consists of DNA that is not surrounded by your chromosome, by your nuclear membrane. So it is known as nucleoid, or this can be called as bacterial chromosome. This is the genetic material that is not surrounded by the nucleus. Here histone proteins are absent. As histone proteins are absent, this DNA is called naked DNA. As this DNA do not have naked histone proteins, it is known as naked DNA, and along with that it consists of a double stranded circular DNA, and this is known as plasmid, or this can be called as a F factor, or this can be called as a sex factor. This is an extra chromosomal DNA, and this is the original DNA called as a nucleoid or bacterial chromosome. This DNA is naked DNA because of the absence of histone proteins, that is nucleoprotein, histone proteins are absent, and this is a double stranded circular DNA which is made up of, which also participates in conjugation. Next, it consists of 70S ribosomes, and magnesium granules and volutin granules are present. Magnesium granules and volutin granules are present in the cell. Next, coming to reproduction, the most important reproduction is binary fission. Andarki telusadi. Binary fission means this is your bacterial cell, it will divide into 2 bacterial cells. So that this is known as binary fission. And next is sexual reproduction, is a conjugation. All of you know the conjugation, that is your sexual reproduction. These are the general characters of your bacteria. In next class we will see the general characters of virus. Okay students, today we will stop here, and in the next class we will continue with the general characters of virus. Okay, thank you students.